Hi everyone, and welcome to the Canadian Poultry Forum. My name is Dr. Stephen Theriault, and I am the CEO and CSO of Cytophage, an up-and-coming biotech company which is changing the way we use bacteriophages to deal with bacterial problems. Cytophage is a company which is heavily invested in the animal health market, with projects in E-tech, Salmonella, Clostridium, and Strep suis, just to name a few. Our goal at Cytophage is to give farmers, veterinaries, as well as pet owners an antibiotic-free solution to bacterial problems. So let's look at where these problems actually come from. When we look at the overuse of antibiotics around the world, all of us are fully aware that this type of use of antibiotics is a huge problem, not only for our food system, but also for our human health. And what we see is around the world, about $9.8 billion, this is in the United States alone, being used to deal with antimicrobial resistance. We also look at how we actually feed our animals. And because we use because 70% of all antibiotics are actually used in our animal feed systems, it causes huge problems for AMR. As well, the WHO predicts that between 30 and 50% increase in the actual costs of dealing with antimicrobial resistance will be seen in the coming decade. So at Cytophage, we basically brought an old solution back to life. So bacteriophage are viruses that only infect bacteria. They are the most abundant form of life on the planet. And again, they only infect bacterial cells. They do not infect animal cells or human cells. Therefore, there is no toxicity that comes actually from using them. And again, I'll add a little bit to that in a little bit. I also want to give a kudos or a call out to Felix Delahaye, who is a Canadian from Montreal, who in 1917 actually determined that bacteriophages could be used to actually treat dysentery during World War I. And he did this, of course, in our Canadian military as well as our allied military. So how do bacteriophages actually work? Well, bacteriophages generally have two life cycles. I'm only going to speak on one of them today because it's the most important life cycle for what we require. The other life cycle, what we call lysogeny, is basically where the bacteriophage can integrate its genetic information into the bacterial cell. We don't like that type of life cycle because it does cause resistance and causes other issues down the chain, especially if you're trying to use bacteriophage as a treatment. We use the lytic cycle, which you can see represented here, and that's basically where a bacteriophage will bind to a bacterial cell, release its genetic information into that bacteria, repropagate itself, building on itself, of course, and then bursting or lysing out of that bacterial cell, killing the bacteria and releasing many more bacteriophage to go out and kill more bacteria. So as a company, Cytophage, again, has about 10 years of independent research. I formed a company around the technology of developing these bacteriophages in 2016, and the goal for us, of course, is to implement a non-antibiotic solution for bacterial infections. And we use heavily synthetic biology techniques or molecular biology techniques to develop our bacteriophages for, again, specific targeting of bacterial cells. So how do we do this? Well, there's many ways that we actually create our bacteriophages. Just looking at sort of four general ways. Again, natural phages can be used when you do find the needle in the haystack for natural phages. And what I mean by a needle in the haystack is a bacteriophage with no toxin genes, no lysogenic genes, and is fully lytic. And again, these phages do exist, so natural phages can be used in that manner. However, most natural phages come with a lot of inherent issues that have to be genetically modified or manipulated in order to make them usable. So in that case, we would use modified phages as well as synthetic phages, where we can actually insert genes and sort of manipulate the genetics of that bacteriophage to get it to do what we actually want it to do. So when we're doing that, again, we use fully synthetic bacteriophages, synthetic phages, or of course modified bacteriophages. And what this allows us to have is a toxin-free bacteriophage. We also remove all insertion genes, so we get rid of that life cycle. And then, of course, we add our own genes to allow for easy modifications. Now, what does this do? This actually gives us a lot of advantages over other companies as well as other groups that are using natural phages. Why? Because we tailor-make our bacteriophages. So we design them. We actually don't discover them. There is no fear of superbugs occurring, again, because these bacteriophages do not harm or cannot bind to animal or human cells. We are extremely cost effective when we develop our bacteriophages because we have unique ways of actually growing these phages. And again, this all is linked back to the genetic constructs and how we can actually grow them. And then of course we only use the lytic cycle, which gives us generally 100% kill of the bacterial cells that we're aiming for. So as a company, we have a lot of product potential, and what we have is 3 independent divisions, one working in human health, animal health, and of course food safety. And all of these are sort of linked together because again, animal health goes into human health, and then food safety of course is all part of that as well. When we look at the animal health programs that we're currently running, again, we have a very robust poultry animal health program where we deal with Salmonella, Salmonella and Clostridium, as well as other poultry problems. But these, of course, are the 2 products that are the furthest along in our chain of getting registered or product approval. So if we look at Salmonella, again, these are products to reduce the colonization load within the actual chickens before harvesting. In animal trials right now, our actual trials are going on and should be concluded at the end of July. And once they're concluded at the end of July, we'll be moving forward with our regulatory process. The reason for that is because we're also conducting all of the toxicity testing that the government requires of us before we can actually get approval. Now, Clostridium, again, is another one of our products where we've created the bacteriophages for the actual bacterial problem. So again, this product is to remove the bacterial load from the chickens because Clostridium, of course, is a lethal model. And from there, we can, of course, move forward in treating the actual disease. Bacteriophage, again, have been generated for this product, and we are conducting our live animal studies at the end of July. So once we finish Salmonella, we'll be starting Clostridium. And just as a highlight, again, we have a lot of swine projects as well as ruminant projects. Just to highlight some of the swine projects, Streptococcus suis, as well as E-Tech, and then, of course, we're looking at mastitis in ruminants. So to give you an outlook of how these animal studies are conducted, again, I'm not going to go through each particular study just because time restraints, but just to give you sort of an outlook, most of it is against dosage as well as toxicity, as well as looking at treatment frequencies. So again, in our particular case, the Canadian government has given us strict guidelines of what they're looking for, and we, of course, are carrying out these experiments as they have dictated. And again, they will be completed by the end of July, and hopefully we can continue moving forward with this particular project through the regulatory approval from the government. When we look at when we look at Clostridium, again, our project is moving forward. We've used a really unique moth larvae animal system, or let's say system, to determine the infectious dose, and we did this in moth larvae as it'll decrease the amount of animals that we actually have to use when we're doing the dosage testing for Clostridium. So it's just a new methodology that we're actually using, and something that actually saves us a lot of time and a lot of animals. So that's very, very beneficial. And we're looking at these projects to be starting at the end of July into the early August, and then of course the phage efficacy studies being done as well during that time. So if we're looking at products and commercialization and moving forward, our closest product for commercialization is the Salmonella product. We are currently in many discussions with many partners about licensing the product and, of course, moving forward for sales and distribution. But again, it's something that we're moving forward with in the sense that we're moving into commercialization with our first product, and that of course is barring the first regulatory approval from the Veterinary Drug Directorate. So from there, I would be happy to answer any questions anybody has, and again, I appreciate the invitation to present today, and I look forward to speaking with everybody.
Well, thank you very much for your presentation, Stephen. If you want to just join me on the...
Oh. Yeah, you're getting my follow-up slides. You can just cancel the talk. There you go. Perfect.
Awesome. So many questions. If you have a question, again, please put them in the question and answer. I have so many questions, but let's start with this one. Thank you for promoting phages. How stable are the synthetic phages, and how do you overcome considering them GMO for application?
Okay, so very stable. Again, that's part of the process that we're developing them. We make sure that they're extremely stable and they act generally like normal bacteriophages. The GMO question is sort of an interesting one. So first off, we are a genetically modified organism. However, if you put the classical term on us, a genetically modified organism that actually affects a human cell, we're actually not a GMO. The other part of it is we're actually treating the bacteria infection. We're actually not putting phages inside of the chicken in order for you to eat them. So the bacteriophage actually aren't present when you're actually eating the chicken or the poultry meat after the downstream sort of mechanism has taken place. So yeah, not a GMO, no issues with GMO. All of the regulatory folks that we've spoken to have not and will not consider bacteriophages being modified as a genetically modified organism, again, because it doesn't infect human cells. So there's no fear of, creating everybody or turning everybody into a sphinx.
Yeah, or whatever other myth goes along with that. For sure. I just had questions like, sorry, I think I might have missed it. I think you said it now, it's to treat, it's not a prophylactic type approach where you apply it broadly, widely.
Well, we can approve it. So again, because antibiotics are being removed from the chicken facilities, we're actually coming in as a prophylactic for that process. Again, replacing antibiotics with a bacteriophage and doing it prophylactically. But we're also looking at treatment as well. So with our Clostridium bacteriophage, we would look for treatment. With our mastitis, of course, it would be treatment instead of preventative. But we can do preventative as well. Yeah.
Okay, and so my next question is, so when you're talking about salmonella, would these phages work with all salmonella, or is it very specific to the serotypes?
So with the genetic modifications that we do, we can increase the amount of bacteria that we can actually tackle. So for us, it's epididymis as well as typhimurium with our product. And as we find that there's other salmonella that are problematic, we'll just create a new phage to put into our product to tackle that one as well. So for us, again, we strive to get as many in the serotypes as we possibly can for the problematic bacteria.
All right. And how do you apply it, if you will? Like, is it?
So we put it through the water system. So we've developed sort of a mechanism where we can feed our phages within the water system. And as the chickens are drinking the water, they're getting the influx of the bacteriophage as well. We also can put it into the chicken food itself as we'll call it a feed additive. And that's another mechanism that we can get it into the chicken's gut. But for us, the easiest path for us is getting it in the water, because it's a very simple process for the farmers, similar to what they used to do with antibiotics. They do the exact same thing with our bacteriophage. So we just find it's a little bit easier for the commercial users of our end product.
And just from a curiosity, from a regulatory point of view, did CFIA say, oh yeah, we know what this is and we know how to classify it? Or is it like, because again, it's novel, that's been my experience sometimes, is not my personal experience, but I've worked with people who are like, well, we've got something new and they don't know how to classify us. So that's the delay.
Happened with the Veterinary Drug Directorate. We've currently had about 4 meetings with them. The first meeting, nobody knew what a phage was. This meeting, they actually know what it is, and they're actually moving forward. And they're very keen on creating Canada as a bacteriophage sort of research hub around the world. So they're very keen on getting these products into the marketplace. In saying all of that, we do have to go move forward. We are classified as a drug, which is difficult in some respects because we're not a drug. We're obviously much different than a drug. We can't do a lot of the science that drugs require, but we're doing all of the science that bacteriophage require. And one of the things that we need to do, which is I always jump back and forth about the necessity of it. We have to do toxicity testing for bacteriophages, which is very sort of wasting of animals. Well, we all know that bacteriophage are non-toxic, but this is just a step that we need to do in the drug application. So my hopes is, as we get the first bacteriophage approved in Canada, we then will hopefully have a guidance document that the government can then use to get the next products and the next bacteriophages on the market without all of this craziness that we had to go through for the last 3 years.
Yeah, I can only imagine.